The beef industry is constantly investigating the most current research and developments that ultimately increases financial returns. It is assumed that nutrition and environment would be the main factors in predetermining fat percentage and muscle coverage in any given beast. Research into these factors has led to the realization that it is predominantly genetic factors that influence increased muscling and regulate muscle growth of cattle. This can be further supported by a study which was recently undertaken by Chantal Coles to determine whether proliferation rates of bovine primary muscle cells relate to live weight and carcass weight in cattle. This experiment conducted by Coles involved 13 6-month-old male calves of 3 different breeds: Angus, as you can see behind me, Hereford and Wagyu Cross Angus, all known to be early maturing animals, with Angus and Hereford both having thick bone structure and the ability to fatten over shorter periods of time. These animals were all grazed on New England perennial pasture, consisting of 3 grasses with good nutritional value and proof of boosting live weight gains effectively. The primary trial species were all male bovine, and samples were taken 30 minutes post-slaughter. These muscle samples were taken from a region of the longissimus muscle to collect primary skeletal muscle cells.
The myoblast cell multiplication rates were tested at different intervals in vitro. These stages included an early stage of 5 to 20 hours and a later stage measured between 20 to 72 hours. As you can see in these graphs, Angus was the breed in both stages which had the highest cell number during proliferation phases. The rate of proliferation, if any, and gene expressions were recorded from 4 specific genes. The 4 genes are all very influential, each having varying roles to play. Specific scientific methodology were used to extract results. These methods included muscle digestion, immunocytochemical staining to determine myogenetic cell populations, measurement of cell proliferation, and RNA extraction. Here you can see the staining with high results in the Angus cells.
A previous study on developing longissimus muscle found that the number and composition of muscle fibers is determined in the developing bovine fetus. Of great interest was another study that found that estimated lean meat yield, or muscle, is a more reliable measure of genetic merit of beef yield. These studies that considered muscle in relation to live weight and carcass weight may have been the precursor that led Coles to research rapidly multiplying muscle cell growth. In conclusion, Coles' research supports that proliferation rates in cattle's primary muscle cells are found to be positively related to live weight and carcass weight. The myoblasts of Angus cattle proliferate approximately twice as fast compared with Wagyu cross cattle. With these results in mind, manipulating skeletal muscles at the cellular level to increase muscle mass and therefore increase beef yield may be an opportunity in the future. Breeding fat cattle isn't all about nutrition and environment, but rather having the right genetic building blocks. Thank you.